Variation in chromosome number and structure. First we'll see variation in chromosome number. This can be due to either addition or loss of one or more chromosome, that is a single chromosome, not a complete set. This condition is known as aneuploidy, represented by the formula 2n plus minus x, where x is the number of chromosome added or lost. For example, as discussed in the previous video, monosomy with formula 2n minus 1 and trisomy with formula 2n plus 1. Variation in chromosome number can also occur when a complete haploid set of chromosomes is present in addition to the normal two sets. This condition is known as euploidy. Depending on the number of sets present, these polyploids can be triploid, that is 3n, tetraploid 4n, pentaploid 5n, and so on. Now, the origin of such aberrations. Such chromosomal variations happen due to random errors that occur during the gamete formation. Non-disjunction is the failure of the chromosomes or chromatids to separate and move to opposite poles during the cell division. When this occurs during meiosis, the normal distribution of chromosomes into the gamete is disrupted. For a particular chromosome, abnormal gametes may be formed that can have either two members of a specific chromosome or none. When these abnormal gametes are fertilized by a normal haploid gamete, it will produce a zygote that has either three members, that is trisomy, or only one member, that is monosomy of that particular chromosome. Now, monosomy is the loss of a single chromosome represented by formula 2n minus 1. The example for monosomy is Turner syndrome, which was discussed in the previous video. Similarly, trisomy is the addition of a chromosome to a diploid genome. The formula is 2n plus 1. Examples of trisomy are Down syndrome, that is trisomy 21, trisomy 18, that is Edwards syndrome, and trisomy 13 or Patau syndrome. These were also discussed previously. Next is polyploidy. Here, instances where more than two multiples of a complete haploid set of chromosomes is found, it is known as polyploidy. Accordingly, the polyploids are named triploid, that is 3n chromosomes, tetraploid or 4n, pentaploid 5n, and so on. Polyploidy is prevalent in plants and is rare in animal species. Polyploids can originate in following two ways. First, by addition of one or more extra set of identical chromosomes to the normal haploid complement of the same species results in autopolyploidy. And secondly, by combination of chromosome sets from different species due to interspecific matings, this results in allopolyploidy. Again, autopolyploids may be formed if all the chromosomes fail to segregate or separate during meiotic division, thus forming a diploid gamete. If the diploid gamete is then fertilized by a normal haploid gamete, a zygote with three sets of chromosomes is produced. Autopolyploids may also be formed by fertilization of an ovum by two sperms that will result in a 3n or triploid zygote. Or autopolyploid may be found if in a cell chromosomes have replicated but the parent cell never divides and re-enters interphase. Here the chromosome number will be doubled, that is 4n. Experimentally, tetraploid or 4n cells can be produced from a diploid cell by applying cold or heat shock to meiotic cells or by applying colchicine to the somatic cells undergoing mitosis. Colchicine interferes with spindle formation, due to which the replicated chromosomes do not separate during anaphase of the cell division, and thus the chromosomes do not migrate or move towards the opposite poles. Autopolyploids are large in size, and that is why these varieties are of great horticulture and commercial value. In allopolyploids, polyploidy results from hybridization of two closely related species, whereas in endopolyploidy, it is a condition where only certain cells of a normal diploid organisms are polyploid. In these cells, replication and separation of chromosomes occurs without nuclear division. Endopolyploidy is the result of a process known as endomitosis. Example, vertebrate liver cell nuclei. Here, multiple chromosome copies occur in the cell where high levels of certain gene product may be required. Genes whose product are in high demand are present in multiple copies in the genome. Example, ribosomal, and tRNA genes. Next, we have variation in chromosome structure and arrangement. Here, first we'll see deletion or deficiency. When a chromosome breaks at one or more places and a portion is lost, the missing segment is known as deletion. This deletion may occur either towards one end, that is terminal, or the segment may get deleted from the interior portion of the chromosome, that is intercalary deletion. The portion of the chromosome that retains the centromere is maintained, and the segment of chromosome without the centromere is lost in the offspring. Then we have duplication. When a portion or part of chromosome is present more than once and is repeated in the genome, this condition is known as duplication. Duplication may occur due to unequal crossing over between synapsed chromosomes during meiosis, or as a result of error in replication before meiosis. In the first case, both duplication and deletion is caused. Third, we have inversion. Here, rearrangement of a linear sequence of a segment of chromosome occurs in reverse order. This segment is turned around 180 degrees. This requires two breaks along the length of the chromosome, and subsequent reinsertion. A loop is formed prior to the breakage. If the centromere is included in the inverted segment, the inversion is said to be pericentric inversion. If the centromere is not a part of the rearranged segment, it is called as paracentric inversion. Note that the length of the arms extending from the centromere is changed in pericentric inversion. Lastly, we have translocation. It is the movement of a segment of chromosome to a new or different location. It involves exchange of segments between two non-homologous or dissimilar chromosomes. Where a part detaches and joins the other non-homolog. Example, Robertsonian translocation in humans.